Okay, so first, good afternoon, and I also want to acknowledge Edouard and Peter for their invitation to participate to this conference. So today I will show you some results of studies I've been involved during the last years at the University of Potsdam. So they are studies mostly on the African climates. So my presentation will be split in two parts. So first, I will show you a lake-level reconstruction in East Africa using Paléoclimatologie related to the African humid period from the Turkana Basin. And so after, I will change completely. I will move from East Africa to Central Africa, and I will show you the result of a calibration transect for a proxy, which is the hydrogen isotopic composition of leaf waxes. So today we have seen a lot of application from Jessica and also from Peter. And so I want to show you some result we produce to calibrate this proxy on modern data for tropical Africa. So first, I will bring you to this region, which is the Kenya Rifts. And I will show you some lake-level reconstruction using shorelines. So when you look at the map of Africa, you can see that most of the large lakes, they are located in this region. So this is East Africa, and this is due to the presence of the East African Rift System, so which is here delineated by large border faults. And we know that since around 15 million years, since the rift initiation, the extension of the rift is creating some basin here along the rift axis. And these basins, they are available to accommodate lakes when the conditions are wet, when there is wet climate condition. So we know that these lakes in the past, they fluctuate a lot, and they will for sure fluctuate in the future. And so we are interested to reconstruct the past variability to better predict how they will fluctuate in the future. And this kind of information, for sure, they will be of a great importance, particularly for the people living there. As you can see, this is a map of the population density in East Africa. And you can easily see that most of the population is concentrated along these lakes. So we have been working in and interested in this region, which is located here in the main map. So this is the Kenya Rift, and here you have the central Kenya Rift, and here you have the northern Kenya Rift. So today you can see that there is only one large lake, which is Lake Turkana, which is located here. And there is also some small lakes to the south. And these lakes are mostly enclosed basins, so they are fragmented. They are not linked to each other. But when you look at the map during the early Holocene, it was completely different feature. You can see that the small lakes, in fact, they were far larger, and the Turkana lake was very large. And in fact, this lake, they were connected to each other. This is what is shown you by this little arrow, this red arrow, which indicates some overflow. So they were connected, and at the end, they were overflowing toward the Nile. So they were really part of the headwater of the Nile. And so they were bringing fresh water to the Mediterranean domain. And also they also participated to the sedimentation of these organic rich layers that are known as sapropels. So in the next slides, I will show you a detailed reconstruction of the Lake Turkana history during the Holocene. But before, I want to show you some information and data from the Lake Baringo, the Lake Naivasha, and the Suguta Valley. So what do we know about the variability of this lake today? So you can see here a dramatic photograph of Lake Baringo. And it's evident that today the lake level is quite high. So in fact, since 2011, following heavy rain during the wet season, the lake level increased by several meters. And the lake surface area, you can see it's increased by 60%. So you can imagine that all the schools, the hospitals, the tourist resorts, which were built almost directly at the edge of this lake, were completely submerged by water. So there was more than 5,000 people displaced during this flooding. And so we may ask, was this flooding event predictable? So when you look at here some historical lake level, this is not reconstruction, they are based in fact on instrumental data from the Lake Turkana and the Lake Nevasha. So the two lakes I've shown you before in the map. So Lake Turkana is 200 kilometers long, and Lake Nevasha is just 20 kilometers long. And you can see that during the last 120 years, they show more or less the same variation. A general decrease since the end of the 19th century and significant variability during the 20th century. And what is very important to see, in fact, in this plot is the scale of the lake level change. This is 15 meters for both of the lake. So 15 meters in 100 years or 120 years, it's a lot. And if you apply this variation to the Lake Baringo Basin, in fact, you will think twice if you want to build any school or hospital or tourist resort directly or close to the level of the lake, because for sure in the future they will be flooded. So this instrumental data here, unfortunately, they just go back to 120 years, and they can only give us insight about decadal variability. But we also want to know what is the centennial and the millennial timescale variability, because it's very important as this kind of long-term variability to better predict the future variation of this lake. So since we do not have any instrumental data for this long-term variability, we need to find other source of information. So when you travel above the northern Kenya Rift, here in the Suguta Valley, you can see here a very prominent feature. You have here a tuff cone, and you can see that this tuff cone is carved by a horizontal line. And this line, in fact, is a shoreline. And it is showing evidence that in the past, this area was covered by a large lake. So now we go into detail into this area to see how this shoreline look like. You have an 8-meter wave-cut cliff. And here you have the level of the highest water level. So we dated this shoreline, and they are around 8,000 years. And so this is striking because today this area is very dry. In fact, it's just covered by a salty puddle. There is almost no permanent lake. But 8,000 years ago, there was a 300-meter high lake. So completely different climatic condition. So now I will show you some data about the Lake Turkana, which is the main site of this study. So before I've shown you some picture from the Suguta Valley, and now we go directly to the north, Lake Turkana. So here you can see the extension of Lake Turkana in the present day, and this is the maximum extension of this lake, because above this level it is overflowing toward the Nile. So the yellow line that you can see here, which are drawn, are in fact paleo-shoreline, which are just mapped by using satellite products. So they are really prominent. So and you can see the scale, 50 kilometers, so these features are quite large. So here, in fact, we're interested by this area, the South Island, because it has very nicely preserved shoreline. You can see them here. And so we developed a detailed survey of this shoreline. So we use high precision differential GPS, which is this little tool. And here you have a satellite image of the shoreline. And so we make some perpendicular transects across the shoreline, and we record very precise elevation for this shoreline. And for selected shoreline, here, we dig some pits, and we extracted some material for dating, because we want to know the age of this shoreline. So here you have the composite profile of our survey in South Island and the elevation, and we mostly dated this freshwater mollusk shell, which are carbonate, and we're using this radiocarbon methods. Unfortunately, we could not date any charcoal because we could not find any, because it's too high energy environment in this shoreline. So here, they are all the sites that we dated. And in the next slide, I show you the results. You have the age, and you have the elevation. And this is all the sites we dated. So with this data, we can make first a tentative lake level reconstruction, and we can say that during the early Holocene, the lake level was high, most probably overflowing toward the Nile. Then we do not have so much information. And then around 5,000 years, the lake dropped by at least 50 meters. So this is only the data from the South Island area. However, we also dated other sites in the western part of Lake Turkana, and there have been a lot of studies before on this shoreline, since I think the 70s. And so you can see that all these points are representing some previously studied site, and we here mostly focus on the South Islands. So now I'll show you all the other ages based on shoreline deposits. So mostly dates on freshwater mollusk shell. So they are showing at least the presence of the lake, or maybe they were deposited slightly below the lake level. We have also other source of information. They are coming from the archaeological excavation. There are these orange circles here. So we collected a total of 102 ages, and you can see that there is quite a lot of scattering the ages, but this is common in lake level reconstruction using shoreline. And so with these ages, we propose a lake level reconstruction. How do we do? In fact, for a given time period, we're using always the highest age as the lake level pass. So for example, here, we trace the lake level because this is the highest age. Also, you will tell me, okay, there is also higher age here, but these one are related to, they have been displaced because of tectonic deformation. I don't show you here the data of the tectonic deformation, but we also use the shoreline to estimate the extension of the rift, and so we had to correct some of the site from the deformation because we are in the rift, so the rift is quite active, and there is some parts which are uplifting, but most of the other parts are subsiding due to the rift extension. However, so we were able to reconstruct that during the early Holocene, during these time periods from around 11 to 8,000 years, the lake was relatively high at its maximum level, and it was overflowing toward the Nile. Then around 8,000 years, there is a drop in the lake, but we don't know exactly what is the magnitude of this drop. Then again, the lake increased to higher level, but we are not sure if it was overflowing, and then the lake dropped at around 5,000 years, at least 50 meters. And in fact, you can see that all the ages which are related to archaeological excavation, which are recording human settlement, meaning that we are for sure above the lake level, they are clustering all along this lake level drop, which is supporting our reconstruction. From 5,000 years to 2,000 years, unfortunately, there is not so much data from the shoreline. We just know that the lake level was low. But in fact, there is a very recent study from people from Duke University. I think they use seismic stratigraphy, and they are showing that during these very dry periods, the lake level was at least 40 meters below the present day. So something somewhat below the slide. You cannot see it, but somewhere down here. So then the lake increased again to slightly higher level until the last millennium to reach the present day lake level condition. So this lake level record is quite in good coincidence with the Paléoclimatic record we know in tropical Africa and with the one we have seen today, which are showing generally wet condition during the early and a part of the mid-Holocene, and then a transition toward arid condition after the mid-Holocene. So this transition is either gradual or abrupt. We've seen a lot of examples, but in this case, we can say that this transition is relatively fast. So what is important with the Turkana basin and interesting is that there have been a lot of studies about archaeology, about the human occupation of the basin. And so now we can compare our climate record with the cultural records in the region. So before 5,000 years, the archaeologists found a lot of very nicely preserved bone harpoons. And at that time, in fact, the basin was occupied by hunter-gatherers who mostly relied on fishing in their subsistence lifestyle. After 5,000 years, it's very different. In fact, there is a drastic change. Also, foraging was still persisted, and fishing was an important part of the diet of the people. This peculiar period is marking the emergence of pastoralist group in the area. And this is well shown by this bovine figurine, which was found on archaeological deposits from this period. And the emergence of this pastoralist group in the area also coincide with the development of megalithic pillar sites in the Turkana basin. So now, when we compare our lake level reconstruction here, which is showing a wet climate during this time, before 5,000 years in the Holocene, and a dry climate after 5,000 years, we see that there is some striking coincidence with the Turkana Basin cultural record. For example, at this transition, there is the apparition of a new pottery tradition, there is the development of the megalith architecture, and there is the emergence of the pastoralism. And you can see that from 5,000 years to 4,000 years, this is the time where you have the highest concentration of radiocarbon ages related to human occupation, which is suggesting that there was mostly probably the highest density of people around the lake. So we can speculate that, in fact, the establishment of a drier climate after 5,000 years in the region of the Turkana Basin may have forced the local herders and their domesticated livestock to converge toward the lake. Also, this lake was shrinking. It was probably one of the last permanent water body in the region, and it would have provided sufficient water and pasture to foster the settlement of these people, ultimately leading to new form of social and economic organizations. In summary, for this first part, I've shown you the reconstruction of the Lake Turkana water level using shoreline related to the African humid period. We identified a major lake level drop of at least 50 meters at around 5,000 years. And we speculated that maybe droughts may have influenced Holocene prehistoric culture in the Turkana Basin. So this climatic reconstruction based on lake level is mostly providing some information about the climate change at a multi-centennial or millennial time scale. And so it will be great if we can combine this kind of information with other climate proxy, for example, with higher time resolution. For example, the hydrogen isotopic composition of the leaf waxes in the sediment, as Jessica and Peter have shown before. So, in fact, now I will show you some new data about this proxy, which is the hydrogen isotopic composition of the leaf waxes in Cameroon. So now we are moving from East Africa to Central Africa, and we are shifting to the geomorphic scale of the shoreline to the molecular scale of the leaf wax. And the hydrogen isotopic composition of the leaf wax, as you have seen before, are increasingly used in paleoclimate studies since the last 10 years at least, and particularly in tropical Africa. However, until recently, there have been no calibration framework based on modern data for tropical Africa, which try to understand what is the relationship between the hydrogen isotopic composition of the precipitation and of the leaf waxes. And so this is the aim of this second study. So before going into detail into these leaf waxes, I want just to give a few words about the hydrogen isotopic composition of the precipitation. So Peter and also Jessica have shown similar diagrams, so I will try to be fast. But what we know is that the distribution of a light and a heavy isotope in the water is controlled by fractionation processes which are occurring during the water phase transition. So for example, you have a transition from here the liquid to the vapor during the evaporation, or during transition from vapor to liquid during the formation of rain or condensation, and then you have fractionation processes. And we know that the isotopic composition of the meteoric water or precipitation is tracking these processes in the water cycle. And the dominant control on these fractionation processes, they are very diverse. There is, in fact, the temperature, the elevation, can be the latitude, the distance from the coast, the moisture source, the rainfall amount effects, the relative humidity. There can be also the transpiration or water recycling. But here we are working in this region, which is here enlarged, and precisely in this area, which is the Cameroon, which is at the border of Central and West Africa. And we know that for this region, the main control on the fractionation process of the meteoric water is what we name the rainfall amount effects. And this is very well highlighted in this plot. Here you have the hydrogen isotopic composition of the precipitation, and here you have the rainfall amounts. These data are coming from this station, from the GNIP network. And you can see that there is a significant negative correlation between the two variables, which suggests that if, for example, you are measuring the hydrogen isotopic composition of a rainfall, you may be able to reconstruct an amount of precipitation. And this is very important for paléoclimatologists. This is one of the climatic parameters they are mostly interested. So now comes the most important question. Can the hydrogen isotopic composition of the precipitation be preserved in sedimentary material? And the answer is yes. And how? By being incorporated into the leaf waxes. So now I come to the leaf waxes. So here you have a nice leaf, and sometimes you may have seen at the surface of a leaf, you may have guessed that there is a transparent coating, and this coating is made of waxes. Here you have a transversal section across a leaf. So you have the cell of the leaf, and you can see a white layer here. And this white layer, in fact, is made of crystals of epicuticular waxes. So what is the function of these waxes? It's to protect the plant from different biotic and abiotic stresses. For example, the temperature, for example, osmotic stress. There is also wind abrasion. So there are several stresses, and so the plant is protecting itself by covering its leaf by this coating. And most of, in fact, the waxes are made of long-chain hydrocarbon compounds. There is the fatty acid, as Jessica has shown us before, and there is also the long-chain N-alkane. So they are very simple molecules, only made of carbon and hydrogen. And if you are looking now at the hydrogen fraction of plants or higher plants, if you look at the gas chromatograph of this higher fraction, you can see 3 peaks here. And they are showing 3 compounds, C29, C31, and C33 alkane. So sometimes you have also the C27 and C35. And these molecules are long-chain, odd-numbered alkane. So why is it interesting to work with these molecules? First, since they do not have exchangeable hydrogen, they are relatively stable to low temperature diagenesis. And so you can find them in very, very old sediment or rocks. They are also very abundant in lake and marine sediments. And their hydrogen and carbon isotopic composition can be measured using this gas chromatograph coupled to the isotope ratio mass spectrometer. And this technology is available since the 90s. So to evaluate the relationship between the hydrogen isotopic composition of precipitation and the hydrogen isotopic composition of the leaf waxes, with a paleoclimate perspective, we have been to Cameroon. So Cameroon is a quite small country, but is often named Africa in miniature because it has a very strong climatic gradient. You can see here the precipitation, which is decreasing from 3,500 millimeter in the south to 300 millimeter. There is also some site where it rained 12 meters per year. You can also see that there is a very strong vegetation gradient, which is following the precipitation gradient, with rainforest in the south and shrub savanna to the north. So in 2009, we have been to Cameroon, and we sampled 11 lake basins all along these transects. And for each of these lakes, we sampled the lake surface sediments and also the dominant plants on the lake catchments. Here you have a view from the northernmost lake. So you can see that the climate shows low relative humidity along the year, so high evaporation, low rainfall, and the vegetation is dominated mostly by grasses, and there is some small shrubs around the lake. In the south, completely different picture. We have a rainforest. The climate is wet, high relative humidity, so low evaporation and high precipitation. And the vegetation is mostly made of trees. So we measured the hydrogen isotopic composition of the long chain C29 alkane, which is shown here on this axis. For simplification, I just call it L like leaf waxes. So here you have the delta D leaf waxes. And we measure also the hydrogen isotopic composition of the precipitation, which I name it W, like water. And to estimate this hydrogen isotopic composition of precipitation, we sampled a lot of what we say surface water, which are groundwater and small stream. And what you can see here is that when you look at these two variables, for the sediments, there is a significant positive correlation, which is suggesting that for this region, we could maybe use this hydrogen isotopic composition of the leaf waxes to reconstruct the hydrogen isotopic composition of the precipitation. So now these values, in fact, are just for the sediments. So I will take the same plots, but now I will add the value of the modern plants. So you have the sediments, and you have the modern plants. So there is the C4 plants. So they are the grasses. They are using the C4 carbon photosynthetic pathway during the fixation of the carbon. And then there is the tree and the shrub in Africa, which are mostly using the C3 pathway. So you can see that there is a lot of scatter in the data. And if you take the plant alone, you will never find any relationship between the hydrogen isotopic composition of the leaf waxes and of the precipitation. So how does the hydrogen isotopic composition of the precipitation is transmitted to the leaf waxes? So I will show you a conceptual model that Jessica also showed before. This model was developed by Sachs et al. And Smith and Freeman. And how does it work? Here you have the precipitation, which for simplification I name W. So this can shift up or down according to the fractionation processes which are occurring in the water cycle. So in the area, we will say, well, it's the rainfall amount effects. Then the water reaches the soil and is taken up through the roots and the woods to the leaf. And you can see that this red arrow here, they are showing you some soil evaporation and plant transpiration. And in fact, the initial hydrogen isotopic composition of the precipitation is what we name de-enriched, is heavier because of fractionation processes related to evaporation. And the plants will use this leaf water to synthesize the leaf wax. And it will apply what we name a biosynthetic fractionation, which is depending on the biosynthetic pathway of the organism. And it is so for the higher plants, this epsilon biosynthetic is constant. So this line is constant. So then we measure the hydrogen isotopic composition of the leaf waxes, which is L, and of the precipitation. So what we can do now is just calculate this isotopic difference. It's a very simple calculation. We make the difference between L and W. If we know that this one is constant, if this one is not constant, that means that we have evaporative processes before the integration of the precipitation isotopic signal in the leaf waxes. So if this difference is large and close to the biosynthetic fractionation, then the difference are very small. There is no evaporative or few evaporative processes. So if along our transects, our environmental gradient we studied, we see that this difference is constant, that means that it may have occurred some evaporative processes, but they were constant. But if this difference is fluctuating, that means it has different evaporative processes along the gradients. So now what I will show you is just the calculation of this difference for the sediment and the plant data. So we will start with the plants. Here you have the difference between the hydrogen isotopic composition of the leaf wax and the precipitation. You can see the scale from minus 200 to minus maybe 80. And here you have the latitudes. In the south, the climate is wet. We are in the rainforest, and in the north is dry. So it's the shrub savanna. So here there is normally along the year low evaporation and high evaporation during the year for the northernmost side. So in fact, you see a lot of variability in the plants data, and which are suggesting that if this difference is not stable, is that there have been some evaporative processes which occur before the hydrogen isotopic composition of the water is integrated into the waxes. And so if this difference is large in the south, is that evaporative processes were the least. And in the north, if this difference is smaller, is that there have been more evaporative processes. And in fact, what you are seeing here is a sampling bias, because when we sampled these leaves, here in the south, we were still in the wet season. And so high relative humidity, low evaporation. So this is why this difference is large. So fractionation processes associated to evaporation are low. But to the north, we were already during the dry season, and so higher evaporation. Now, when we look at the sediment, where we have a complete different picture. In fact, the sediment seems not to record these evaporative trends that we are seeing with the plants. They show almost a flat line. So why is it the case? We need to think about when does the leaf wax develop. So first, when does the plants are developing in a dry ecosystem of tropical Africa? They are growing when there is the water. And so this is a climate diagram from Kano, which is in Nigeria, which is almost at the same latitude of the driest site we investigated. So you have here the months and the precipitation. So you can see that there is a very small rainy season of several months. And I show you here a vegetation index, which is the normalized difference vegetation index. It's an index which derived from satellite products, and it is a good index for the vegetation cover. And what this index told us is that the plants in the region of Kano, they are growing only during around 2.5 months. And when are they growing? They are growing when there is the water. So if they are growing when there is the water, this is the time of the year when the relative humidity is at its highest level in this very dry region when we take the annual average climate. And if this high relative humidity, evaporation is at its lowest. And what do we know about the leaf waxes? When are they growing? There is some greenhouse experiments which have shown that, in fact, for the trees, the leaf wax take place early during leaf development. So here, this is the time of the leaf development. If we apply this observation to this region, then we may propose that the leaf wax are growing when the water is available and when there is the least evaporation in the ecosystem. So what, in fact, the sediment is telling us is that there is a vegetation effect which is counteracting the effect of the evaporation all along these gradients. And we need to say that the sediment is, in fact, an optimal substrate because it is integrating thousands and thousands of plants which are living around the lake basin. And it is integrating these plants at the scale of, at the spatial scale, but also along the time, because the lake surface sediment is not recording one day as the plants we sample. It is recording at least 1 year, maybe 5 years. So it is, in fact, providing us the hydrogen isotopic composition of the landscape. So if we have this relationship which is flat and which suggests that there have been no additional fractionation processes related to evaporation, or if there have been such processes, they were constant along the gradient, then we can use the hydrogen isotopic composition of leaf waxes with confidence to reconstruct the hydrogen isotopic composition of the precipitation. Imagine that there have been an evaporative trend. Imagine that this square were following the value of the plants. Here we sample just one time during the year. Then it will be very difficult to use this lake surface sediment data to estimate the precipitation isotopic composition, because first we will need to correct them from the evaporative processes before applying them as a reliable proxy for the isotopic composition of the precipitation. So to finish, the integration of our new sedimentary data from Cameroon with previously published sedimentary data from Europe shows that the hydrogen isotopic composition of the leaf waxes are excellent proxy for the hydrogen isotopic composition of the source water. And this across very large environmental gradients, which are representative for the entire northern hemisphere. So in summary, for the second part, I've shown you that the hydrogen isotopic composition of leaf wax and alkene from tropical plants can be highly variable, and they are reflecting different fractionation processes which are related to evaporation mostly. However, the lake sediment are smoothing out the individual plant variability, and they result, in fact, in a stable apparent fractionation between the leaf waxes and the precipitation, which is suggesting that the hydrogen isotopic composition of sedimentary and alkane can be used with confidence to reconstruct the isotopic composition of the precipitation. What next? When you look at the present-day map of where the lake sediment core have been analyzed in tropical Africa for their hydrogen and carbon isotopic composition in the leaf waxes, you can see that they are all in East Africa, where the large lakes are. I don't speak about the marine record, just about the lake sediments. Our transect was made in this region, which is quite far away from this site. So we went back to a lake that we sampled in 2009, and last January, with an international team, with some colleagues from Cerej and also some Cameroonian partner, and using this platform, we covered the 100-meter deep lake Barom Bimbo, and we recovered in continuous 20 meters of finely laminated sediment. So now what we want to do for this project, we want to apply this biomarker, this hydrogen isotopic proxy to reconstruct the hydroclimate for this region with a special focus onto the African humid periods. So to finish, I want to acknowledge all the collaborators who participated with me to this project, and particularly Manfred Strecker and Dirk Sachse for their continuous support during my stay at the University of Potsdam. And I want to thank also the Dutch Forschungsgemeinschaft and the Alexander von Humboldt Stiftung for having funded this project. Thank you.